bacteria and viruses. So bacteria and viruses are the part of microbiology, where we cannot see these living organisms with our naked eyes. So here these are the one which are responsible for many positive and also negative impact on the life of other living organisms like human beings, animals and also plants. So here these are the one which are having some specific characters. So in the course of its classification, they have been separately kept. Neither they have been included as a part of animals nor as a part of plants. So here let us discuss some of the important questions which are related to bacteria and viruses. So let us come to the questions. Question number 1. Unicellular organisms that divide by binary fission are the given options: A. Viruses, B. An independent group between viruses and bacteria, C. Fungi and D. Bacteria. So here the question is related to unicellular organisms. So in the microorganisms, so unicellular organisms are the one which are made up of only single cell. That is the entire body is made up of only one cell, and that single cell will perform all the functions related to an living organism. So in the given options, we can consider the first one as a viruses. Actually viruses are considered to be acellular in their nature. So acellular means which does not have cellular organization. So that means they does not have cell wall and they does not have plasma membrane. But how should we consider them as living organisms means, because they have the genetical makeup as like that of other living organisms with the help of DNA and or RNA molecule. So but as we are considering these viruses as acellular, so it will not be the part of unicellular organisms, so we can leave it. So some other group of organisms are called as mycoplasmas. So mycoplasmas are also considered as a microscopic organisms which does, but they does not divide by binary fission. And the third group are the fungi. In fungi, some of them are microscopic in their nature, but here in fungi some of them are unicellular. At the same time there are some multicellular organisms also. But these fungi will participate in different types of reproductions like fragmentation, with the help of formation of the spores, that is asexual process. So fragmentation by vegetative process. At the same time they will participate in the reproduction by formation of gametes, that is a sexual process. And there are some fungi which will participate in vegetative process by formation of buds like yeast, which will perform the process called as budding process. But nowhere we can say that the fungi group are performing binary fission. And one more group of living organisms are bacteria. So bacteria are unicellular in their nature, and bacteria are made up of a single cell, that is unicellular in their nature, and the cell is covered with cell wall, and inside the cell wall the plasma membrane will be present. So here this bacteria will perform binary fission. So how does this binary fission will occur in this bacteria means? So let us discuss in brief this character. So let us take a bacterial cell. This is a bacterial cell which is having cell wall, and inside which we can see the presence of plasma membrane also, and the nucleus will be present. Actually we cannot say it as a nucleus because it is not covered with nuclear membrane. That means nuclear membrane is absent, nucleoplasm is absent, and nucleolus is also absent, but the entire nucleus is represented by only circular naked double stranded DNA molecule. The first thing what happens is this nucleoid, we can consider this as, actually this nucleoid structure will first elongate and gradually it will divide into two parts. First it will elongate and in the next step it will divide into two equal parts. By the meantime, when the nucleus is divided into two parts, by the meantime what happens, a small constriction will develop in the cytoplasmic contents exactly nearly at the middle region where the mesosomes are present, and this constriction gradually move towards the center, and this will also move towards the center. When both of them will meet at one point, the bacteria cytoplasmic contents are divided into two equal halves like this. And by the meantime, the cell wall will also form the constriction exactly at the position where the cytoplasm have been formed the constriction, and that constriction will also gradually move towards the center and divide the bacterial cell into two equal halves. Likewise, one bacterial cell is converted into two equal halves by a process called as binary fission. So this is the general mechanism of, we can say, the common method of reproduction which occur in the bacteria during favorable conditions. So out of the given options, we can say that bacteria is a unicellular organism which will perform this type of binary fission by vegetative process. So from the given options, option D is the correct answer. Question number 2. During Graham's stain, the given options: A. All bacteria gram positive or negative take crystal violet stain. Option B. Only gram positive bacteria take crystal violet stain. Option C. Only gram negative bacteria take crystal violet stain. And option D. Gram positive bacteria lose this stain after alcohol treatment and take red stain of safranin. So here this is a question related to the staining process. Here this concept of staining was developed by Graham scientists, so it is called as Graham staining process. So in the Graham staining process, actually it is a process to differentiate the bacteria, where we can say that some of them are considered as a gram positive and some of them are considered as the gram negative bacteria. First, all the bacteria which have been to be differentiated, they have to be kept in crystal violet such that all the bacteria will take up or absorb the stain in the form of crystal violet. And when this bacteria kept in crystal violet, as I said that all the bacteria will take up the stain, and after that it have to be treated with alcohol or acetone. So when it is treated with alcohol and acetone, what happens, gradually some of the bacteria will lose that stain. Some of them will lose the stain, and at the same time some of the bacteria will retain that stain. The one which will lose the stain are called as, that bacteria are called as gram negative bacteria, whereas the bacteria which will retain that stain are called as gram positive bacteria. So this is a one to differentiate the bacteria depending upon the cell wall material that is present in them. So here from the given options, we can say that all the bacteria will take up the gram stain, either it may be a gram positive bacteria or a gram negative bacteria in the first step. So from the given options, option A is the correct answer. Question number 3. A cubical packet of 8 cocci is called. The given options are A. Sarcina, B. Spirillum, C. Staphylococcus, and D. Vibrio. So here this is a question related to the shapes and the arrangement of the bacteria in groups. So depending upon the shapes and the arrangement pattern, so there are actually basically some characters are there like cocci, bacilli, vibrio, spirillum are the types which we can see in the bacteria. Cocci are the spherical bacteria, whereas bacilli are the rod shaped bacteria, vibrio are the comma shaped bacteria, spirillum are the spiral form which are coiled like a spring like structure. So here in this one, when you come to cocci, so depending upon the number of spherical shaped bacteria, so there are different types are there like monococcus, diplococcus, tetracoccus, staphylococcus, streptococcus, and along with that one there is one more called as Sarcina. So from the given options we can say that Sarcina contains a group of 8 spherical shaped bacteria like this. So 4 will be appear to be like this, and the remaining 4 will appear to be on the present on the outer side like this. So one more fourth one will be like this. So 1, 2, 3, 4, 5, 6, 7 and the 8th one will be there. So here all the 8 will be arranged in the form of a 8 cocci bacteria arranged in the form of cube, or we can say cubical packet of 8 cocci are called as Sarcina. So from the given options, option A is the correct answer. Question number 4. A chain of spherical bacteria is called streptococci. When spherical cocci are found in grape-like irregular arrangements, they are called. The given options are A. Staphylococci, B. Sarcina, C. Palisade, and D. Streptobacilli. So here this is also the question related to the spherical shaped bacteria. Spherical shaped bacteria are generally called as cocci. Plural it is cocci and singular it is called as coccus. So here as we are saying it as monococcus. If 2 are there, diplococcus. If 4 are there in the form of a chain like structure, we call it as tetracoccus, and a chain with many such spherical bacteria are called as streptococcus. Streptococci we can say plural, okay, streptococcus or streptococci. At the same time, if this spherical bacteria are present but in the form of irregular arrangement like this, so it may appears to be like a bunch of grapes or whatever we can consider, but the arrangement should be in a irregular pattern without any specific sequence of these spherical shaped bacteria. So if like this a cluster or a group of spherical shaped bacteria are present, so we call this as a staphylococci. So in the given options, B it has been given as Sarcina. Sarcina means 8 spherical shaped bacteria in the form of a cubical packet, and Palisade is a tissue which is present in the leaf, and Streptobacillus means bacilli means spherical. It is not spherical shaped bacteria, but it is a rod shaped bacteria. And from the given options, Staphylococci, that is option A is the correct answer. Question number 5. When a spiral bacterium has only one curve and comma like, it is called. The given options are A. Spirillum, B. Vibrio, C. Bacillus, and D. Spirochete. So here this is also the question related to the shapes of the bacteria. In this one, first one it has been given as spirillum. Spirilla means it is a spiral shaped bacteria, so which will have little bit curve like structures like this. And Vibrio is the one which is having comma shape, like it appears to be this type of structures. Comma shaped bacteria will be there. And next one is given as Bacillus. Bacillus is a rod shaped bacteria, so like this. Then last one is given as Spirochete. Spirochete is the one in which the spherical arrangement will be more like this. And one more thing here, it is it will be showing more number of coils here, and but in the question it is given as only one curve should be there. So here one curve should be there, and it appears to be a comma like structure. So from the given options, Vibrio, that is option B, is the correct answer. Question number 6. Free living, non-symbiotic, aerobic, non-photosynthetic, nitrogen fixing bacteria in soil is. The given options are A. Azotobacter, B. Rhizobium, C. Clostridium, and D. Anabina. So here it has been given as the bacteria should be a free living one. Free living one means actually bacteria which are not associated with any other type of living organisms, or which does not have symbiotic association with any other type of living organisms. We call them as free living bacteria, and that's what it is given as. That should be non-symbiotic also. So here non-symbiotic means actually before that non-symbiotic we should have a little knowledge about what do you mean by symbiotic association. If two different types of living organisms are associated with each other, and that association should be a mutual beneficial association. That means both the living organisms should get benefited, or we can say the first organism should get benefited from the second one, and second one the same time should get benefited from the first one. And that type of association we call it as symbiotic, and both of them should not cause any type of harm to the other partner. And next one is aerobic. Aerobic means which will perform respiration in presence of oxygen, and non-photosynthetic. So we know that photosynthetic means which will perform photosynthesis and prepare its own food material by trapping carbon dioxide, solar energy, and water. So here it is given as non-photosynthetic means it should be an parasite, or it should depend upon the host for its nutrition. And nitrogen fixing. Bacteria. So nitrogen fixing means the higher plants does not have that capacity of taking or absorbing the molecular nitrogen and converting it into the useful form of nitrogen, because they actually for that function they require an enzyme called as nitrogenase enzyme. That nitrogenase enzyme is present only in the lower organisms, like in some of the cyanobacteria and in some bacteria. So here this capacity is there with only the bacteria and the cyanobacteria. Let us take the examples like Azotobacter that have been given in the options, and the second option is given as Rhizobium bacteria, and the third option is given as Clostridium, and last one is given as Anabina. So here Azotobacter is the one which is free living, non-symbiotic, aerobic, non-photosynthetic, nitrogen fixing bacteria. Whereas if it is considered to be Rhizobium, Rhizobium is the one which is symbiotically associated with the roots of legume plants, and Clostridium is is not showing that character which have been given in the options, and of course Anabaena is cyanobacteria, which is also considered to be the photosynthetic one. Likewise, some characters are not shown with some these three group of organisms which have been given here, that is Rhizobium, Clostridium and Anabaena. So from the given options, Azotobacter is the one which is free living, non-symbiotic, aerobic and non-photosynthetic nitrogen fixing bacteria. So from the given options, option A is the correct answer. Question number 7. Pili or fimbriae are surface appendages that never help in locomotion but help in. The given options are attachment, B conjugation, C pellicle formation and D A and B, that is attachment and conjugation. So here this is the appendage which is present on the surface of the bacteria, so which are considered to be the structures which are present outside the cell wall. So these are surface structures or surface appendages, so which are concerned with the two functions. What is for example, if one bacteria is having that pili or fimbriae, so they will help in attachment to the surface. Or there is one more type of pili are present, those are called as sex pili or sex fimbriae. These are the one which are there with the bacteria, and they are also useful for attachment but not to the surface or any substratum, but it is useful for attachment of one bacteria to the other bacteria. So what is the purpose of attachment of one bacteria to the other bacteria means? So actually here the bacteria will perform three types of genetic transfers. Those are called as transformation, transduction and conjugation process. During the third one, that as I said, that conjugation process, consider one bacteria is present. One bacteria which is having this conjugation, which is participating in the conjugation, or we can say the two bacteria which are participating in the conjugation are called as conjugants. And here the one of the conjugant which contain this long elongated hollow structure, that is called as sex pili. So that will help in attachment of one bacteria to the other bacteria like this. And inside the bacteria, this is a genetic material, that what we call it as a nucleoid, and this is a plasmid part. This is nucleoid, and so here we can call this as F plus bacteria, and this is called as a F minus bacteria. So during the conjugation process, what happens is the plasmid part will undergo replication and forms into a copy or one more copy of this plasmid, and this plasmid part will be transferred from this F plus bacteria to the F minus bacteria. So here, as during the process it is acting as a tube like structure, so we can call this as a conjugation tube. That is, here the sex pili which is having hollow inside it will be helping in transfer of this genetic material from one bacteria to the other bacteria, so we can call it is acting as an conjugation tube. So here the pili are concerned with the two functions, one is called as attachment and other is called as a conjugation process. So from the given options, option D is the correct answer. Question number 8. Mesosomes in bacteria are considered equivalent to mitochondria. What is correct for mesosomes? The given options are A. They contain respiratory enzymes like cytochrome oxidase and dehydrogenase. B. They are infoldings of cell membrane to increase surface area. C. They are attached to nucleoid to provide energy during binary fission and help in nucleoid separation and septa formation. and D, all of the above. So here bacteria are prokaryotic organisms. That means they lack mitochondria, which is considered to be the respiratory structure. But here initially when the bacteria structure was discovered, or it had been come to known, so the scientists had found that some sort of inner foldings have been formed by the plasma membrane. This is a cell wall structure, and somewhere here in the this is plasma membrane, where we can see the inner foldings somewhere. So these inner foldings are known as the mesosomes, this one. So the scientists, when they have been come to know that this type of inner foldings were present, they consider that these are the one which contains the respiratory enzymes. So that means respiratory enzymes means the enzymes which are being used in the respiratory process. So like cytochrome oxidase and also the dehydrogenase enzymes. That means respiratory enzymes are present in the mesosomes. And of course, as I said, that these are the infoldings of plasma membrane or cell membrane. And when they are forming the inner foldings, can we say that so we can see clearly that the surface area of the plasma membrane will increases because of this inner foldings. The surface area of plasma membrane will increases. And here this is a structure which will be helping in the process of binary fission, or we can say that this is a site where the binary fission initiation will be taking place during the vegetative process. And not only that thing, whatever the energy which is required for binary fission, because binary fission needs energy which is an active process to be considered. So whatever the energy that is required for the binary fission will be provided, and that not only it is providing the energy which is required for binary fission, but also it is helping in the nucleoid separation and the septa formation. That is the wall which is separating the two bacterial cells. Here whatever the option that have been given in A, B and C, all the three characters are being exhibited by the mesosomes. So from the given options, option D is the correct answer. Question number 9. Nitrosomonas and Nitrobacter oxidize. The given options are A, nitrate to nitrogen, B, carbon dioxide to carbohydrates, C, nitrogen, that is N2 to NO3, and D, ammonia to NO3, that is nitrate. So here the two bacteria that have been given, those are Nitrosomonas and Nitrobacter. So these two are considered as a nitrifying bacteria. In the process of biological nitrogen fixation, this nitrification process will be present. That is a nitrification process. Here, as we know that 5 steps are there. So in this in the process of nitrification, what happens is the ammonia which have been formed as a process of ammonification process, or simply to say, in the first step what happens is the molecular nitrogen is fixed, and that had been converted into the organic nitrogen form that is utilized by the plants. And when the plant dies or an animal dies, the organic nitrogen which is present in the particular living organism will be converted into ammonia by a process called as ammonification process. And after ammonification, whatever the ammonia that is there in that organism, so that have to be converted into nitrite and into the nitrate molecule. Ammonia have to be converted into nitrite, and nitrate have to be converted into nitrate molecule. So here Ammonia, when it is converted into nitrite, is done by nitrosomonas, and nitrite, which is converted into nitrate, is done by nitrobacter. So in the given options, that is given as ammonia to nitrate is the one where the nitrosomonas and nitrobacter are participating in the process. So from the given options, option D is the correct answer. Question number 10. Which bacteria shows pleomorphism? The given options are Clostridium, B. Acetobacter, C. Salmonella, and D. all of these. So here this is a characteristic feature which is exhibited by some of the bacteria called as pleomorphism. So this is also being related with the shapes of the bacteria. That is generally the bacteria, majority of the bacteria will have one specific shape. Either they may be spherical, rod shape, spiral, or the vibrio, comma shape. But there are some exceptional cases where they will change their shape according to the environment in which they are existing. Second one, depending upon the nutrition that is available. So here some bacteria change their shapes depending upon two reasons. One is the environment in which they are existing. Second one is depending upon the type of nutrition that is available to them. So here sometimes they may have rod shape, may be converted into a chain of rod shaped bacteria. Otherwise they have been converted into vibrio shape, or sometimes they may be converted into spherical or spring like structure. Likewise they may be changing their shape depending upon the two reasons, that is the environment and the nutrition that is available to them. So likewise that character is called as pleomorphism, and that character is exhibited by a bacteria called as Acetobacter. So from the given options, option B is the correct answer. Question number 11. The bacteria that cause botulism is Clostridium botulinum. It is, the given options are obligate anaerobe, B. endospore producing, C. exotoxin producer, and D. all of the above. So here botulism is the character that have been exhibited by a bacterium called as Clostridium botulinum. So Clostridium botulinum is a bacteria which is causing a disease called as botulism, that is a spoilage of the food materials. So this is an obligate anaerobe. Obligate anaerobe means which survive only in absence of oxygen. So anaerobe means generally which will respire anaerobic type of respiration, that is in absence of oxygen it will perform respiration, and obligate means it cannot survive in the presence of oxygen, that means it cannot perform respiration by taking oxygen, and it will produce some special type of structures during unfavorable condition called as endospores. So endospores are resisting structures which will form a thick wall around it, and that is the one which is a protective sheet formed by this bacterium, and whenever the favorable conditions are retained, that endospore wall will be liberating these internal structures and forming into a new bacterium, and at the same time the Clostridium will produce some chemicals into the food materials or the organic materials in which it is surviving. So that chemical is called as exotoxin. Exotoxin is a toxin, means toxic material, means which is an poisonous material that will be released into the food materials or the surrounding environment in which this Clostridium botulinum is present, and that chemical will actually that chemical is called as botulin chemical. That chemical will spoil the food materials. So here it is an exotoxin producer. So in the given options, it is exhibiting the characters like obligate, anaerobe, endospore producing, and also exotoxin producer. So from the given options, option D is the correct answer.